very important for one thing, which is the color of the slide, and especially the dark as well as dark but yet bright magenta backgrounds that you're going to be seeing in the slide. It's based on scientific research evaluating attention span for online users. But the most important reason for me up there, you know, in that section of your screen, is to keep you paying attention and to facilitate the learning of the information I'm going to be sharing. It's based on a principle that was really started to be uncovered in the late 1990s. It's called retinotopic mapping. And this is an area in the brain that literally, when the brain is experiencing, and our body and our visual system is experiencing 5 specific earthbound things, it goes straight back to the visual processing center. It doesn't go all the way around the 7 different synapses. It goes straight to the visual processing. And that's because our survival and reproductive health are literally dependent upon it. And those 5 things are faces, tools. Just think about early man needed to know about tools. Also, letters, numbers, and the most important and the most primary is what you see in your peripheral vision. It's the reason why we have more rods in our eyes than we do cones. And so consequently, you're going to be seeing me move throughout this presentation to jog you awake, hopefully not, to keep you paying attention, and to facilitate your retinotopic mapping so that you're able to best enjoy this presentation as well as facilitate the learning, because I have a lot to share, and most of you, this is going to be new information. So the second thing right now, actually the third, is look at the title, Skin, Organ of Photoreception and Immune System Response. Well, since the late 1990s, early 2000s, the lighting design, architectural, and interior design communities have really started paying attention to how light gets in to the eyes. And we're so accustomed to understanding photoreception. Well, when it comes to skin, we're not talking of just purely ocular. We're talking about extraocular photoreception. So whenever you are doing a presentation, or you're speaking to a client, or you as a stakeholder, building owner, or even homeowners that are watching this presentation, you have to understand that light is a stimulus, a biologic stimulus, and it's getting in 2 very important pathways. Our eyes, primarily for circadian entrainment as well as neurohormonal response, as well as the skin. And it is also connected to the circadian system. Wow, who knew, right? Simple light. Okay, so let's get started. Okay, so the skin. I like to think of it as the organ of first response. Now, we have defined in medicine 10 pretty much, normal systems, so to speak. You've heard your medical provider, or in biology class, they're speaking about the circulatory system, endocrine system, nervous system, et cetera, et cetera. But one of the most important systems that, again, since the 1990s, in addition to the circadian system, the skin, or the integumentary system, sorry for that mispronunciation, is also vitally important. And in medical terms, the skin is called integument. And it's the largest body, the largest organ in our human body, and it's approximately 16 to 20 square feet laid out flat. And let me tell you, the first time I saw dermis laid out flat, I was taken aback. It was incredible. Weighs pretty heavy, 8 to 11 pounds. And you have to understand that the skin is alive, literally and figuratively. It grows, it produces waste, and it sheds itself, just like your dog. Just like your dog sheds hair, well, the skin sheds skin. And about every 4 weeks, new skin appears at the top. And every day, every hour, we are shedding approximately 30 to 40,000 dead cells every hour, and that amounts to a lot, about 1.5 pounds of skin a year. And over a lifetime, oh my gosh, I think some estimates are like 63 to 65 pounds over a lifetime of just dead skin. But the important thing about skin as an organ, it has to do something. So what it's doing for our survival is pigmentation. And it's designed to protect against and regulate UV radiation, ultraviolet radiation. And the skin color and thickness is not uniform across the entire body and face. And all you have to do is just look at your elbows or the pads of your feet or the heels of your feet and your abdominal region. You just look at the different skin layers and you have different valleys and habitats for that skin and the critters that live on that skin. We'll get to that in a minute. So whenever I start a program, I always like to think, what is the who, what, where, and when type of a deal? What's the question? So I'm going to ask what's the importance of the skin? Well, it's there to regulate temperature, both internal and external temperature, to regulate it so that the body isn't totally freaked out when the temperature drops down. And this can be witnessed in goosebumps, shivering. Uncontrollable shivering could be also a sign now of the COVID-19. But it's there to make sure that the skin is a homeostatic balance. Basically, it's on even par with everything that's in its surroundings. The skin provides shape, and it also is an avenue to eliminate waste. And waste being the sweat and perspiration, particularly those areas of the skin, under the arms, under the neck, the buttocks region, between the legs, the interior thighs. Those are capable of producing mucus as well as secretion in terms of sweat. And sweat is very important for the survival of that skin. You'll see in a minute why. So why? I mean, why do we have skin? Well, why do we have it and how does it work? Well, it's working on the principle that it adapts to the environment to survive and reproduce. And those of you that have attended some of my classes in the past realized that survival and reproduction is the one thing that we all living beings need to do in order to survive and reproduce and to have our lines and our genes go on. And how does the skin do this? Well, it's because of its structure. It's watertight. In other words, water does not get in unless you cut yourself. And it has a microbiome diversity that acts as a shield. And this is the part that I'm not going to have too many queasy photos, I promise. But you have to understand that there are things, living beings on your skin, on your face, in your eyebrows, and in your lashes right now. And they're there for a very, very good reason. And that purpose, simple: protect, regulate, and defend. And keep in mind that skin is part of the immune system. So protect and defend, you know, it has insulation, the adipose layers, insulation and adipose layers. In other words, everywhere that you accumulate fat, there's extra skin there as well. It's because something inside is extremely important that if it gets injured, well, you're in a lot of trouble. The temperature regulation, it's there for translating the sensation of pain, pressure, and temperature. And no, skin cannot tell by itself if something is wet or dry. It needs the brain to interpret that information. So the only three senses that the skin is able to translate to the brain directly for interpretation is pain, pressure, and temperature. And we actually have various sensors within different layers of the skin to specifically do just that. And it's for our evolutionary fitness, UV protection, and destruction. And one of the key ways that we understand this is through vitamin D synthesis. Now keep in mind that every cell and every tissue throughout the body has a vitamin D receptor. Vitamin D is, we used to think was just a vitamin. Well, it's no longer that. It's a critical, important, predicated on the fact that it's there to protect us and as well as provide us immunity. And the thing about the way the body works is that it's almost like a car, you know, a gas pedal and a brake, gas, brake, gas, brake. And so what one action is when one action is engaged, something else is being let loose. In other words, you're stepping back, and then it instantaneously goes like this. It's almost like a perpetual motion machine. Folate, vitamin B, is another one of those such things that works in opposition to vitamin D synthesis. It has a conflicting response to light, and it's a very important reason why we understand this from an evolutionary perspective. So when we look at skin pigmentation, the difference is by latitude. It's evolutionary in origin, vitamin D synthesis. And the reason why it works with folate and vitamin D is for reproduction, particularly in women. Women at this period, from anthropology files and studies, have shown that women seem to dominate. And when women close to the equator were not getting protected from the UV rays, their skin started becoming more pigmented to prevent flattening of the pelvis and preeclampsia, which if you're an emerging species, you need to reproduce. And so simply by looking at the line of the equator crossing through, you can see where some of the pigmentation is the most heavily pigmented with color. And the interesting thing is that right now, we used to think that there was 5 to 7 different skin color types, and the number of scales, scientific scales, were promoted for years. Here are the 5 types of color. And that is really starting to be questioned right now because of the varied nature of our skins. And this is showing that we truly are becoming a melting pot. But the skin, no matter what our pigmentation is, the skin is very important for DNA. 70% of the proteins encoding genes are contained within the skin itself. So in other words, the skin needs to be protected, and you cannot survive without a full covering of skin, not for long anyway. And also the skin is circadian and trained with photoperiod response. And we'll talk about that a bit later, as well as opsin-based process for light phototransduction. And those of you that are very familiar with the health and light concept of light understands that phototransduction is how we get the light from our eyes back to the IPRGs, through the brain, through the pathway into the SCN, and then all of the neurohormonal responses are occurring. But the thing that we need to understand is that the skin, as part of its protection and defensive mechanism, has 10 to the 31st, 10 to 1 combination of different organisms living on our body. It's designed to protect against viruses. And viruses, I mean, right now they're 10 to the 31st. Someone estimated that the number of viruses that are out there are more than 10 times the number of stars in all of the galaxies that we understand to be existing right now. And the microbiome is charged with one thing. It's a first-line immune system defense, and it is also providing a response. So let's look at the layers of the skin. We have 3 basic layers. The first is the epidermal layer, and you can see it here, taking a look at the slide, the image down at the bottom. The epidermis doesn't look like much. It seems like not that much is going on in there. Well, a lot happens because there's no blood cells there. So in other words, it's there for an evolutionary purpose. If you get cut, it's not going to bleed as bad in terms of that area. But the melanin, through the combination of existing inside of melanocytes and keratinocytes and Langerhans are what is keeping us in tune with the environment, as well as part of the immune system. And you can think of the keratin, because that's part of the integumentary system as well, as keratin, as hair and nails. That was once a living fraction of us, and it's now hardened. And this is what protects our fingertips. Our hair is for energy conservation and management. In other words, thermal control. It's the reason why we have hair on our head. And also the melanocytes, they're 95 present in the epidermis, but keratinocytes outnumbers that, and they have an immune function. And part of that function is wound healing and the involvement of 2 critical genes, P53 and NP53. And you might be familiar with that. These are genes that are associated with melatonin, and melatonin, as we know, is the primary reason for melatonin is a tumor suppressant. And secondary, third, fourth down the line, it's the melatonin, the hormone of darkness, and it helps us sleep. But the primary function is a tumor suppressant. The other thing about that top layer is that it communicates with the nerve cells. it starts to send information down deeper so that the brain can respond. It's anticipatory, very similar to the circadian system is anticipatory. The stratum corneum. Okay, so this is the area that is at the very top of the epidermis. There's 5 layers to it. The dead skin, desquamation is up at the top. And that's actually a 1-month cycle from when the new skin cells are starting down in the keratinocyte level. It takes about a month for them to come up to the surface and then flake off and just lay there until something removes them or they naturally fall off. And the microbiome resides here, and it's an ecosystem of 1.8 million square diverse habitats. So in other words, we have different microbes living under our arms, between our legs, behind our ears, in our mouth, all different places that we have skin. You have different combinations of microbiomes, and they're all there for a reason. And then finally, or next level, we have the dermal layer. And this is the layer that communicates with the environment. It's a connective tissue, hair follicles and sweat glands. Fibroblasts are the cells that represent this area, particularly those that are involved with collagen, elastin, and these are the fibers that are light responsive. They're in the visual reflected area of light. And we know what the action spectrum of that is. And collagen really creeps up in there to some of our everyday SPDs of the light that we have in our offices. So it's important to understand that when we occupy an office and don't regularly get outside, or we're constantly under UV or regular LED lighting, collagen is paying attention to it. And we have the hypodermis layer, subcutaneous layer. This is fat and connective tissue, and BAT, brown adipose tissue. And the interesting thing about that is that this is the good type of fat. It is very sensitive to cold environments and very sensitive to light right here on our neck, behind, and up in this upper supraclavicle area. And this is the area that if we get the right light at the right time, this is going to help control the fat. Down here, now you can't see it, but it's on my abdominal area. So you want to keep the rooms as cold as possible, as well as light as possible to contain the fat in the area that it's supposed to be. And here's the kicker. Take a look at the image that I have right here. This is how deep light penetrates through those 3 primary layers. And even those 5 layers of the epidermal, the stratum corneum, it gets in UVC, UVB, UVA, and the visible all the way down, all the way down. And when we get into 630 nanometers, it's way down. And when we definitely are in the IR and IR near, it goes all the way down to the muscle layer. And so here's another image that I need to point out to you. So take a look from the left to the right. We're looking at the blue, the bright blue image. That's where 405 nanometers reside. It gets into the skin. So I know a number of manufacturers are exploring, and some are even actively marketing, the use of 405 for occupied spaces. Well, here's something that I know one of the primary researchers did not know anything about in terms of the microbiome and how it's impacted by 405. So do your research, folks. So let's look at skin receptivity of light through the placenta. Well, okay, this is a study that was fielded several years ago in veterinary science. But I would not be surprised if in the coming decade ahead, we really start connecting the dots and start understanding this from a human perspective. And basically this study, it was actually done in 2013, shows that daily doses of light with a minimum of just 3 lux of full spectrum, including UVA and UVB, so true full spectrum, was enough to impact fetal development in the first trimester. It impacted the neuronal numbers, the oxygen demand, postnatal vascular development, in other words, your veins, and the development of normal vision, all of that in the first trimester. So do you want to take a guess who they experimented on? Well, obviously not humans, but on mice. So even through their hair and through their very fat bellies, light getting in is impacting the fetus in this case plural, that they're carrying for normal vision development, and only in the first trimester. So we also have to look at light as not just the UVB, UVA, and UVC as carcinogen, but something that is potentially phototoxic. And this is for light, LED light. It could also be, depending upon how close you're sitting, it could be halogen light, any numbers of light. But we have phototoxicity. And what this means, it's a dermal response to light interaction with various drugs, food, and reproductive hormonal changes. And right now we know that we have about 200 plus known drugs, including antidepressants, antibiotics, cardiac and diabetes medicine, that respond negatively to photo illumination in spaces and literally high levels of various wavelengths of light. And it's not easy to know, say, for example, if you're a lighting designer or an architect designing an ICU, or even just a normal cardiac care room scenario in a healthcare situation. So you need to understand what their drug protocols are to be able to really figure out where you are locating your light luminaires, as well as what is the light benefit to that person's skin, not just to their eyes, if we're figuring circadian relevance, but to the skin. And of course, phototoxicity can be derived from foods, including celery, carrots, limes, when rubbed on the skin and exposed to light. So I'm going to show you some pictures, and these aren't too bad, so I promise. Okay, so here is a dermal response that is literally representative of most very severe type of dermal responses. And I'm not even going to get into all of the medical names and all of the medical diseases because there's a ton of them. And this is something that is, and down below we have melasma, which most women will experience this, particularly after a hysterectomy, or if they've had the complete hysterectomy, in other words, where the uterine, the fallopian tubes, and the ovaries had been removed, as well as after going through the pause, the menopause. And it's a response to hormonal imbalances. And no matter what you do and how much cover up you use, it is always there. Believe me, I know, I have one right there. So anyway, this is something that lighting designers need to understand, because depending upon the occupied spaces and the individuals that are in that space, you may be exasperating some of the conditions. And so this is something just to think about, especially when you're doing your due diligence and you're doing design development. Now, sun allergies are common, and this is common across all populations, again, and with all skin colorations. It's a red rash, but it diminishes after exposure. In the presence of UVV, here's the question: will sun allergies be prevalent? In other words, when we start introducing ultraviolet visible light, the 405 nanometer, into occupied spaces, will allergies be prevalent? As far as I know, and I've looked through a blue moon of studies, and by the way, I have a white paper on this topic, which will be coming out in the next week or so, that has about, at this point, I think it's like 63 or 67 different citations. And I've been through probably 100 more, and I could not find anything in terms of that relevance. But so the third bullet point is WWUD. What would you do? If you're an architectural or lighting design professional, what would you do? You know, how are you going to address this? How are you going to sell this to your client and have the architectural leads buy into this and not have it VE'd out? So here's something else that we've all heard about: skin, light through closed eyes. A lot of people in some of my sessions, when I talk about this, they're like, wow, you mean I'm sleeping at night, but I know when the sun is starting to come up, and it may not even be above the horizon line that I see, but my body's paying attention to it? Yeah, exactly. What we do know for sure is that it's about 1.5 millimeters thick. The purpose for an eyelid, the thinness of the eyelid itself, is not yet defined. Could it help speed, the movement of pupillary light reflex? Is it a reception of early pre-dawn light to ensure cortisol awakening response? Those of you that are familiar with circadian science understand that CAR is very important, because this is basically when the circadian rhythm goes in, it juxtaposes with the sleep rhythm, and it starts to awaken us to get ready for the day. And it's very important that your eyes pay attention to this, even while you're sleeping. And here's a study from RPI, and Mariana took the lead on this, and the study was done in 2012. And the experiment was this. It was very simple. 2 60-minute light exposures, and it suppressed melatonin by 36% on the first 60 minutes, 45% in the middle of the night, and at the end of the night, basically when CAR, cortisol awakening response, was really kicking into gear, 56% at the end of night, because that's what you want to do with melatonin at the end of night. You don't want it to express anymore. You want to start dampening it down. But the big thing here was that the circadian phase delay was by 17 minutes after initial exposure, and by the time the end of the night, it was 71 minutes after second exposure. So that's something that we don't want to do on purpose, because we're going to be circumventing the natural inclination. But this just goes to demonstrate that our skin is really adapted to our environment, and it is dedicated to protect and defend us no matter what. And skin, there's a light connection, and it's actually called photoprotection. And this protects against generation of free radicals and ROS. ROS, if you read any of the papers that I've outlined in this white paper, it's reactive oxygen species, and it's basically unstable oxygen molecules, and when it's left to build up, it damages DNA, RNA, and it leads to cell death. It's something that you don't want to have happen. Internal conversion of DNA proteins and melanin to absorb UV energy, that's part of photoprotection. Photochemical process converting UV photons into heat, that's really important. That's the main secret of UVB 295, specifically about vitamin D. And the reason why it's not going to hurt us is because it's ultra fast. It destroys the DNA in our skin on purpose, but it does it ultra fast, 10 to the 15th in femtoseconds. And I don't think I'm pronouncing that right, but there's no time to react. And that means if it reacts, then it's going to start another cascade of something called NP53, which is the DNA repair process. And we don't want that to kick in exactly when it is destroying DNA. And that's where melanin comes in. Probably all heard about melanin. But and you also have heard about 5 opsins that are in the eye. Melanopsin, rhodopsin, panelin, et cetera, et cetera, et cetera. Well, melanin and melanopsin, yeah, the same thing that we have in our eyes is in our skin. And that was discovered in 1998 in frog skin. Who knew? That what's so important, and what we've been hearing about for a decade and a half now, is actually in our skin, and it's been there all along, and we've known about it since about the same time that we first found out about the connection for the suprachiasmatic nuclei, the master biological clock, and the IPRGs. Skin light receptors. So here's how it works. Light penetration. It's wavelength specific, and it's purpose specific. So take a look down at the bottom of the slide, and the colors represent various wavelengths. And you can see on the far right, the white. It goes all the way down to the muscle layer. And the bottom most of that cube section is muscle layer. And that's the reason why so many of the NIR techniques and photodynamic medical therapies are using NIR, because it goes down to the muscle layer. And you can even purchase some of those handheld devices, and some of the catalogs, you can buy it for muscle relief and pain relief. We have the opsins. They're also the light receptors. They're sensory proteins, and there's 5 of them that we know of in the skin. And in 1994 is when we first realized that they were outside of the eye. Example is OPM3. These are found in these work specifically for informing the brain and the testes. Yes, I'm women, I'm not going to go there, but yeah, that's true. And all these opsins modulate 6 hormones. It converts light into electrical signals. And the keratinocytes on the epidermal layer, these are very important as well, because this is where a good portion of the opsins reside. And DNA is also found in this layer. P53 and P21 involvement. And the reason why we're concerned about that is because P53 for tumor suppressant and P21 is playing a part, according to some papers, in regulating the amplitude and frequency of the SCN, the master biological clock. And rhodopsin. This is one that we've all heard about. It's what, visual purple in the eye. It's what's in the rods. Well, it's also in our skin. And in our skin, it's very sensitive to the blue-green region of visible light. So if we look down at the slide, you can see how far it goes all the way down into the dermal area, and it's also into the subcutaneous area, just barely missing that. And rhodopsin, OPN2, is ocular, and we also have it to mediate dim and bright light. Well, it does the same thing in our skin. And on the skin, it's dim sensitive, and it really responds quite nicely in dim, dark environments. It's a protective mechanism for us. And light receptors. Here's that same image blown up so you can see it a little bit better. And I've also included in the right-hand side of the slide an ultraviolet spectrum that's showing the combination of UVC, UVB, UVA, and UVV. UVV is something that we are really starting to pay attention to now that, again, I mentioned that some of the manufacturers are realizing the disinfectant properties of the 405 on surfaces. This is the distinctive difference. It's on surfaces. So here's another slide showing how deep this penetrates. Something to think about. So the skin, it's always in a war. It's battling to protect. And the reason why we have the skin and vitamin D and the folate channel and destructive pathways and getting vitamin B and D into our skin is for energy conservation. It happened about 1 to 2 million years ago. We needed to be able to conserve energy to be able to, again, reproduce and survive. And folate, vitamin B versus vitamin D. Remember, gas pedal, brake type deal? It's about destruction and protection. Vitamin B will be able to allow then, when you destroy vitamin B, folate, you then allow that for vitamin D synthesis to occur. And it's evolutionary fitness, again with women. Folate protects women for successful childbirth, and it is very instrumental in preventing preeclampsia, in other words, preterm birth. And cancer prevention is what the modern science is now talking about and demonstrating. And it has absorption peaks, 2, in fact, just to make sure that we get it right. That's how much evolutionary perspective is on this, and the evolutionary protection mechanism. 2 absorption peaks, 1 in the UVB range and 1 in the UVA range. There hasn't been any studies that I've found for folate in the UVV range, and I think that needs to be done. Very important, particularly in office environments, teaching environments, and healthcare and long-term care, where the predominant occupants in terms of gender are most largely women. So here's the process with vitamin D. This destroys DNA on purpose. It's called photomelanoma-metabolism. Yes, and I did have to practice saying that, but it's been years that I have the opportunity to practice that, because this is a topic that I get asked all the time. So wait a minute, wait. You're telling me that DNA is destroyed? Yeah, that's how we get the hormone vitamin D. It's a process. You have to destroy the epidermal skin, the DNA that resides in here. Remember I said 70% of the skin has a lot of the, 70% of our protein encoding DNA is, residing right here. So when the sun at 295 hits the skin, it's going to convert the D2 that's in our skin, and it's going to allow it to go 12 hours later on a pathway to both the liver and the kidney. Once it gets into the kidney, Then it converts to vitamin D3, and that's the hormone. And this is the beneficial aspect of vitamin D. It's providing a very strong immune response. It's now proven multiple times to be instrumental in the initiation and progression of disease. And it's particularly important for older populations. And older populations, you have to understand, is two concepts: older biologically and older chronologically. Old age starts to happen at about 35 biologically. Number of our systems and our faculties start decreasing very, very specifically. But chronologically is when we start thinking. And I like to think that old age is like 15 years older than my current age. And I'm not going to tell. So anyway, but photomelanoma metabolism, it's an immune response. And it, again, also involves P53 and P53, which is the repair pathway for 53 and DNA repair pathway in the skin, and P21. And P53, by the way, is particularly sensitive to 405. So here's a representative from a major study, and I'm going to talk about that study in just a second. And you can see in the upper section, it's listing where folic acid, which is vitamin B, is going to take the absorption most readily. And there's 2 spikes. And down at the bottom, it shows the photomelanoma metabolism, where lightweight comes in, 295, represented here by just the standard 300. It'll go all the way down to the bottommost layer, the epidermis. And it literally starts synthesizing into vitamin D2 there. That then is converted in the liver and the kidney about 12 hours later into vitamin D3, which is the hormone. And this is our disease protection. And this is what we need to keep up throughout all of our lives to be able to remain as healthy and as active as we are. Folic acid degradation also occurs, but it occurs at 360. And remember, it's happening at the same time. And even though that's a 60 nanometer span difference, it's happening in, 10 to the 15th or whatever it was back there in terms of the slide. I'll have to go back and check. But it's happening ultra fast. So it happens almost spontaneously. And these are 2 really good studies. And I left these up here in large print because if you really want to understand this concept a little bit more in depth, check out these 2 studies. And again, all of this presentation today will be on my website probably in the next week or so. And so you'll be able to watch it at any time you have the opportunity or pass it on to your colleagues. So let's look at skin, 405 nanometers specifically. And one of the best studies I found was fielded in 2018. And this is where they finally got out of the idea of just looking at bacteria and mold, et cetera, on surfaces and started looking at 405 impact on human skin. And they were actually able to get volunteers. They were males, they got the volunteers, and they had to lay exposed with their derrieres out there under the light. And it equivocated to about 2 hours in the Texas sun. And then they did punch biopsies of their human buttocks. In other words, they put an instrument on and pulled out a slice of skin. And they found that there was no ROS, reactive oxygen species, for either 385 or 405, the 2 wavelengths that they were looking for, but that genes associated with photoaging and inflammation, there was a significant increase of that problem at 6 hours for 405 nanometer. So I'm questioning those manufacturers. Have they thought about recommending this product in a classroom, in a health environment, in a long-term care facility where occupants will be staying for 6 hours at a duration? What are the consequences? The other thing you have to understand about 405 is it's readily promoted on the internet at Amazon. Even at $27.95, you can buy a 405 wavelength machine because it's designed for photo curing of resin. And ask the beauty professionals out there some of the problems that are associated with curing nail polish in the lights after you get a manicure and they shine the lights on you. Well, that's 405. Think about it. So this is the thing that I'm not speaking about UVA, UVV, but I'm asking the researchers out there, please pay attention, because every one of the studies I've come across, specifically dealing with 405 and 385, are speaking to the fact that it's not well understood. It's only partially understood. And 405 in some cases has a morphological change on bacterial surface. And this is important. So even though the bacteria lives on our skin, it's there for a reason. Do we really want to kill the bacteria on our skin? You know, those studies that I have in this white paper, numerous surface lab studies, but there's not many human studies. And I have to give credit to Mark Ray at LRC. They did a study that came out in 2020, and they used UVA 366 in a NICU, a neonatal intensive care unit. But they protected the occupants in blankets and barriers. In other words, they put drapes over top of the isolates. And this study, as well as the others, you know, that I have looked at, you know, cited the need for more studies, and that we need to understand more the surface finishes and material fading, because just like in the RPI study, as well as the other studies I've come across, all of them that have utilized 405 in occupied spaces have reported significant fading in some of the areas of color, and the combination of color or the reflectivity of color. So let's talk about the ooey gooey stuff. I promise I won't scare you too much. Microbiome is also determined and called microbiota, depending upon who you speak to. But it is comprised of bacteria, fungi, viruses, and arthropods. Yeah, oh my. Outnumber skin cells 10 to 1. And they live in colonies, and that's driven by skin topography. Dry, moist, and sebaceous. Dry, pretty much like this area. Moist underneath your arms, sebaceous, your glands in your face, and on some regions of your back, sebaceous glands, sebum. And the microbiome, microbiota, it rapidly changes in response to the environment, very rapidly. In other words, it's going to respond. That's what an immune system is supposed to do, respond quickly to protect the host, us. And the host immunity modulates skin cutaneous immunity. So if I'm sick, I'm going to be more likely to fall prey to some of the problems that are out there floating in the air. The viruses, and now that COVID-19 is on the top of everyone's mind, the disease, the SARS-CoV-2 virus themselves, this is airborne, it's aerosolized when we speak, when we cough, when we sneeze, and this is something we have to contend with. But the microbiota colony disruption also impacts the host immune system. Remember I talked about the gas, the brake, going this way. So if the host, in other words, if I'm sick, I'm more likely to catch something from the microbiome that's out in the atmosphere. Oh, by the way, the microbes, arthropods, bacteria, fungi, and viruses, and that live on my skin, they don't get inside and harm me unless there's a break in the skin. And if my immune system's not up to snuff, that's where the problem comes in. But the other thing, the other important aspect of microbiome is that they educate our dermal T cells, because we also have immune cells living on our skin and in the different layers of skin. Again, an immune response. So this is one little hairy creature, and it's living, you know, on top of us right now. And it is Staphylococcus epidermis. It's specific for body location. On the skin, it's harmless to healthy individuals, and surface transmission. That's where we have a problem. So that's the rush, I believe, that we're seeing so many UV light treatments, UV light applications for occupied and unoccupied spaces to get rid of staph and strep and MRSA and some of the other bacteria that normally live on our skin. But once they get deposited on the surface, that's where they become a problem, particularly for immune compromised individuals. So the thing you have to understand, and we're almost getting to the bottom of the slide presentation, then we'll take some questions, is the microbiota. You have to understand the 3 types that we have. Out of all of the microbiome that live on our skin, there's just 3 types. So take a look at the 4 pictures. We have a nice honeybee down there and a hippopotamus. And by the way, you see that pink and that red around their eyes? Well, that's their sunscreen. When I was in Africa, I got an opportunity to look very close to one, and it's incredible, the skin of a hippo. And then we have some oxen, and then we have a tick up at the top. So you have to define the type of microbiota that live on our skin. Commensal, they're symbiotic relationship where one benefits while the other is not harmed and doesn't biologically benefit. So you want to take a guess at which one it is? Well, if you guessed in the upper right-hand corner, the oxen and the egrets sitting on top, that's commensal. That's an example of commensal. In other words, the bacteria that are commensal, they sit on our skin and they help us. They ward off any type of viruses that come in. They ward off any type of problems that are approaching us. And they quickly jump to help in the wound healing process. We have mutualistic, where both biologically benefit. Take a look at the pictures, see which one or 2. Yeah, honeybee and the hippo, those are examples of mutualistic behavior. And our microbiota is the exact same thing. They both biologically benefit. And this is an example of some of the arthropods that we have on our skin that I will talk about. And the parasitism, only one is harmed. In other words, this parasitic, and that's up at the top, a tick. So basically it lives off of its host. Ticks, as you know, consume blood, and in that process, they will deposit their saliva, and that saliva is toxic to human, and that's the reason why a rash will develop. And if it's left to go out through the blood system, the blood supply and the pathways, then it leads into a full-scale response and or a disease formation and progression. So here's an example of how we can use the microbiome to help us determine who's going to have a preterm baby, preterm birth, PTB. So this is a 2014 study, and it's part of the Human Microbiome Project. And preterm birth is anything that's less than 37 weeks. And it looked at the microbiomes, the microbes particularly associated with PTB, Lactobacillus crispatus. In other words, some of the lactobacillus that's in yogurt, et cetera, they also live on our skin. And this study looked at 1,527 live birth pregnancies, and they followed 600 in depth to full-term delivery. And what they found is something interesting, that in the future, medical professionals are going to be able to determine if a woman is in the first trimester, more likely, in other words, the first few weeks of her pregnancy, is if she is able to carry a baby full term, or she's going to be set up to be possibly a PTB. And it also has to do with vitamin D deficiencies, and this is common among reproductive age women. So if we look at, and I'm sorry I don't have a pointer on my slide advancer here, but under first trimester is to the far left, and look at the long pink slidey things up at the top where it says preterm birth. That is something that you do not want to see. That is a microbiota associated with preterm birth. The second area, the second trimester, you can see it's still there. And then it disappears and it goes down 100% into the third trimester, and in postpartum, it's still there. And that's because the microbiome changes as the pregnancy progresses. And we can follow that in medicine. Microbiota, light and humidity. So we're going to look at something that may be kind of creepy, but dead skin cells and arthropods. So this is what dead skin cell on the left looks like, and what it's supposed to look like when we look at it from our perspective of just looking at our arm or our face or whatever. But we don't see, we're not able to visualize the teeny tiny dead skin that lays on top. But there's two critters here. I want you to take a look at these two. One is supposed to live on our skin. The other, not so much. One's not parasitic, it doesn't bite, but the proteins in its composition, in its body makeup, are allergenic. That means the immune system response for those that are immune compromised may have allergic rhinitis, in other words, allergies, skin, nasal discharge, etc. One of these two critters are an arthropod, not an insect, and it has a 90-day life cycle for females. And they're called dust mites. They're less than 1 millimeter. And they survive on moisture from warm air, and light exposure dries their habitat. And the upper left-hand corner is just a typical allergic rash that we get from dust mites, but no harm is done. And which one is the parasite? Well, that's the critter in brown. That is a bed bug. That is something that if we look upon close inspection, you can, if your vision is good, you can see it. That is something that we don't want in our beds. The dust mites are there for a very important reason. Excuse me. Demondex mites. We have two types. The first lives in the hair, particularly the eyebrows and the eyelashes, and the second lives on the face. They're mutualistic. They both benefit. We benefit from having those there. This is what they look like. They live in our eyebrows and in our eyelashes, and depending upon the humid conditions and how much light we get at night is going to depend upon how bright and how robust the colony of the Demondex mites are. And they're part of the normal flora, microflora, in other words, the microbiota of our body. And they're primarily found, this type, in facial hair, again, eyebrows and lashes, and they're photonegative. They only come out from the hair shaft at night to reproduce, and they can go as far as up to 1.5 inches away from the hair shaft. And they're microscopic, we don't see them. But they're there, we now think, science now thinks, and from what I'm reading, is that they are there for a very important reason, for virus protection. And it's an emerging theory, but you have to stop and think. Here they are coming out. This is a microscopic view. And you can see them down at the bottom of the red shafts, and they're starting to emerge to come out. These are now thought to be feeding on viruses, viruses that live on our skin and have no harm, and viruses that are falling on our skin from airborne aerosolized viruses. So we don't want to unintentionally kill these Demondex mites. It's not necessary. The only time that we have a problem is with patients with rosacea and some of the other skin-borne conditions where an overabundance of Demondex mites is lining up with the potential for rosacea as well as the increasing progression of that disease. So in summary, the skin is both primary organ and immune system first line of defense. It demands our personal consideration whenever we specify lighting, and especially germicidal UV light and UVV. The microbe is alive, and it's responsive to our ambient environment, including light and temperature and time of day. And as architects, designers, and lighting designers, we control this in the built environment. So we need to be understanding that what we are doing impacts the occupants. Other than just the circadian relevance, it impacts everything. And UVA disinfection, it's an emerging theory not applicable for widespread use. This is something that we have to start understanding. The same can be said for UVV. So here's the last, it's a series of six questions that you, particularly as lighting professionals, need to ask manufacturers and their reps before even considering specifying anything that is UV content for occupied spaces. because we don't even know what we don't know. And there's just too much at stake. Here's the 6 questions. So ask your rep, what human studies have vetted your product? And have you looked and examined the impact on the product on age groups, skin types, gender, health status, reproductive viability? All of that matters. We know that from circadian research, hardcore circadian research, but we don't know this for UV, GUV rather. What are the specific areas not recommended for the use of your product? That's really important. So those manufacturers that say, yeah, go ahead and use it in classrooms. Well, classrooms could be anything from preschool to secondary school to college level. Different age groups, different needs for light on the skin and in the eyes. What end user occupational programs and follow-up guides has the manufacturer put into place to ensure compliance, that the company and what they're offering is not going to be misused? What happens if the product is installed and the janitorial staff does not check it regularly, or the automated controls are not properly vetted and maintained and observed on a regular basis to ensure that individuals are not unintentionally harmed? How is your product measured, excuse me, for product stability? What is the fundamental frequency generating the wavelength? That's very important. Is it phosphor coated? Is it a true fundamental of frequency? What part of the LED energy is phosphor converted? Particularly important for hybrid systems. What fail-safe measures are innate to guard against unintentional misuse by the occupants? And is your product, this is a big one, is your product insured? In other words, monetarily insured against fading, reduced durability, premature degradation, and failure of the Wiesenbach test. Okay, so, excuse me, until it's time for a drink of water. That's your homework. Check out what the Wiesenbach test is. So, thank you for your attention. Thank you for questions coming in, and I'm going to take a drink of water. And Andrew, if you can go ahead and pass me some questions, I'll be more than happy to address them.
You bet. Give you some questions while you grab a drink. Not surprisingly, we have a number of questions that have come in. We'll start out with the first one that references that study down in Texas. Basically, the question here is, can you discuss the intensity levels of the 405 nanometers in that particular study in the sun versus the standard product being used as a visible light disinfectant? That comes from Philadelphia, PA.
Okay. There are no corresponding studies, and that's part of the problem. So we're not getting, well, let me use this as an example. There are a number of products out there for photodynamic therapy where back about 5 or 6 years ago, someone came up, some lab came up with the idea of using the microbiome and changing the microbiome, particularly the bad microbes that are inhabiting the gut of somebody with IBS or IBD. And so what they devised was taking an LED light that was 405 on a digestible camera pill and allowing it to go into the intestinal tract. And they found that there was no problem. In other words, it killed the organisms that they wanted to kill. In other words, those microbiome that wasn't supposed to be in there. And the patients all seemed to be very happy with the results. Well, almost immediately, another lab came out and they wanted to document to see if in fact it was safe. And they did an extensive course of study, immediately published the results, and came out with, yes, we tested this from top to bottom and we could not find any problems associated with the 405 inside the gut intestinal tract. And this is part of the problem that we have with 405 right now. We don't have very many consistent tests across the microjoules that we're using, the wavelengths, the timing, the duration, the population. Most of the tests are on surface decontamination. So this is the problem that we have. And so unfortunately, you're just going to have to read the study and do your due diligence, just like I did, to get an exact answer to your question. So hit me with another one.
Got it. Apparently, in various theatrical lighting applications, UV 365 is used for a blacklight effect. Can you speak to any potential harms or dangers associated with this application?
Yeah, blacklight effect. There was a study in Japan a few years back where the light was used that, in other words, in this nightclub, they did not utilize the correct UV light, and they installed it in the nightclub, and there were a number of participants in this nightclub, in other words, the dancers and the band, that were harmed because of the UV penetration into their skin. And so this is the problem that we have. We say that we have a regulatory body, and in photobiology, in the photobiology committee, you know, we do our best to put out, you know, the safeguards and outline and say when and how and where to put this, but we don't really make that much of an effort on the manufacturing side and the user side to pay attention to this. At one point, and I don't know if it's still in current use, anything that was germicidal had a different type of socket, like when we were using T12 lamps, and you weren't able to do that. I'm not familiar with, we have that with LED, particularly interchangeable LEDs. So next question.
Next question is a clarification. Can you speak to UVV as opposed to the other UVs in terms of wavelength?
Okay, UVC is the most powerful for disinfection. We typically on Earth do not receive very much, if any, because of the penetration of the atmosphere and the wavelength coming through. And if we do, it's very minimal. UVB is where we have significant damage to our skin. This is where a lot of the problems occur, from sun tanning. 295 is where vitamin D begins its photomelanoma metabolism process. And this is where we have a lot of the bleaching that comes in our environment from fabrics, et cetera, and UVA, particularly in the area of about 360 on up is where we start seeing significant increases. And the interesting thing is that 405, that aspect of the visible, the UVV, is very powerful as compared to 385 in some cases, but it's hit or miss. And we don't have all the information yet, and we don't know if 405 can safely, through all age groups, through all skin types, through all infirmaries and immune conditions, be safe for occupied spaces. And that's my caution. If we can do it, if we can verify it, hey, I'm all for it. But you know, we need to know more information. Next question.
I hope I get this right. The question is, are all ROS bad? Is that enough information for you?
Yeah, that's reactive oxygenated species. It all depends. And this is an area that you really have to be specific. And you have to be specific in terms of what it is in relation to. Is it the gas pedal or is it the brake aspect? You know, sometimes you want the ROS to do its bad thing and sometimes you don't. So it has to be answered and addressed very specifically. So next question.
So how do we as lighting designers address demands for circadian/stimulus/antimicrobial lighting in regards to long-term health effects versus short-term improvements? So basically, how do lighting designers deal with this?
Okay, basically you bite the bullet and you buy a spectrometer and you ask for samples. The reps will be happy to give you a sample, and you test and you test and you test. And you make sure that your meter is accurately calibrated to read the SPD, and you start asking questions of the manufacturers. And if the manufacturers are not going to provide that, you want to know why. And you're looking at the light source. Then you're also looking at the optics. You need to see how the light source from the straight LED, and and the combination of LEDs, if it's a multiple diode light source, you need to see how it is in its virgin state, and then once it's inside the luminaire, and see if it's distorted. Once you understand that, you'll get a better handle of what it is that you're specifying and why you are specifying. And just above all, just remember that it's not just about circadian stimulus, it's about photoprotection in the skin. That's what it's about. We have to understand that difference. Maybe one last question, two last questions.
Really, I only have one last one. Okay. It's been a request to revisit the light penetration slide. And I think the right answer, correct me if I'm wrong, is that you will be offering this presentation on your website so they can perhaps go back and view that. But if you want to flip to it now, that's fine. But
okay, let's see if we can do this.
While you're doing that, do you have a website that people can use to find this presentation in about a week or so?
BenyaBurnett.com. My partner,
okay.
Benya, we work together, and it's BenyaBurnett. And let me see if I can find that slide. Okay, here's two of the penetration slides. And so they're from different sources. One was basically from the internet, the other from a study, and I have that cited in the white paper, the one on the lower right-hand side, the ones that are in purple and red. And then the other is just a grab from the internet. And so basically you can see the top layer is the epidermis, and then in the middle is the dermis, and then the subcutaneous layer that's where a lot of the action happens. And you can see the blue lines, the blue squiggly lines, those are blood vessels. And then some of the little objects that are in that upside down slice of pie, basically shape, are the receptors that we have for pain and touch and temperature. And the slide that's on the right-hand side, you can see where, again, this is an artist representation of how far that the ultraviolet, visible, and infrared go down into the skin. The UVC is there for specific purposes. UVB, again, it doesn't need to go much deeper than where it does on the epidermis, because the primary reason for UVB is for vitamin D photomelanoma metabolism and to destroy folate B to be able to do that gas and break reciprocity. Visible light, this is where the circadian aspect comes in. Visible light, particularly in the lower 400s, is very important for rhodopsin in the skin. And then once it gets up to the 450s and beyond, it's very important for the ocular photoreceptivity, for our circadian entrainment and the circadian system response to keeping it on track so that it aligns correctly and it is keeping all of our neurotransmitters, neurohormones in the way that they were originally designed. Okay. So,
wonderful.
Any other questions?
You know what? I think that wraps it up, Deborah. We have we're at quarter after the hour.
Oh no!
Yes.
Okay.
I would personally like to thank you for joining us today. I thought it was a great, very interesting, very informative, best webinar. I really liked the format. Thank you so much for joining us today.
Okay.
I'd like to thank everybody from the Rocky Mountain area as well as across the country for joining us today.
Yes.
Again, for more information on any of our upcoming events, please join our website or check out our website at iesrms.org. That concludes today's presentation. Until next time, stay safe, wash your hands, and have a great afternoon. Thanks a lot.
Bye, everyone.